The hydrogen peroxide model is a simple and effective approach for evaluating the antioxidant effects of factors that may be useful in treating age-related macular degeneration or similar neurodegenerative diseases. The oxidant agent, hydrogen peroxide, is delivered in one single pulse, making it faster and simpler to perform than models in which the hydrogen peroxide treatment must be repeated for several days. Because the number of cells seeded influences the oxidative effect of the treatment, it is important to count the cells carefully before their culture. Demonstrating the procedure will be Thais Bascuas, a postdoctorate from Gabriele Thurman's laboratory. For conditioned medium preparation, 28 days after transfection with PEDF, GM-CSF, or both, seed the human ARPE19 cell line T75 flasks at a 500,000 cells per milliliter of complete medium concentration. When the cells reach approximately 80% confluency, replace the supernatant with fresh complete medium and collect the new supernatant after 24 hours. To purify the histidine-tagged GM-CSF and PEDF proteins, centrifuge the supernatant to eliminate potential remaining cells and set the sample aside. Next, add 30 microliters of nickel NTA solution to a 1.5 milliliter tube. After centrifuging, discard the nickel NTA flow-through and wash the pellet 2 times with 200 microliters of 1 times incubation buffer per wash. After the second wash, resuspend the pellet in 40 microliters of